Immunoglobulins. Basic structure of immunoglobulin. We have about 6 structures of immunoglobulin and they are 1. Heavy and light chains. All immunoglobulin have 4 chain structures as their basic units. They are composed of 2 identical light chains, 23 kD, and 2 identical heavy chains, 50-70 kD. 2. Disulfide bonds. Inter-chain disulfide bonds. The heavy and light chains and the 2 heavy chains are held together by inter-chain disulfide bonds and by non-covalent interactions. The number of inter-chain disulfide bonds varies among different immunoglobulin molecules. Intra-chain disulfide binds. Within each of the polypeptide chains there are also intra-chain disulfide bonds. 3. Variable and constant regions. The variable regions are responsible for antigen binding. The constant regions are responsible for biologic functions like complement activation and binding to cell surface receptors. When the amino acid sequences of many different heavy chains and light chains were compared, it became clear that both the heavy and light chain could be divided into 2 regions based on variability in the amino acid sequences. These are the light chain, VL, 110 amino acids, and CL, 110 amino acids, heavy chain, VH, 110 amino acids, and CH, 330 to 440 amino acids. 4. Hinge region. This is the region at which the arms of the antibody molecule form a Y. It is called the hinge region because there is some flexibility in the molecule at this point. 5. Domain. These are the 3-dimensional images of the immunoglobulin molecule which show that it is not a straight molecule, rather, it is folded into globular regions, each of which contains an intra-chain disulfide bond. These regions are called domains.
Light chain domains, VL and CL, while the heavy chain domains, VH, CH1, CH2, CH3 or CH4.
6. Oligosaccharides. Carbohydrates are attached to the CH2 domain in most immunoglobulin. However, in some cases carbohydrates may also be attached at other locations. 3 different types of mechanism of immunoglobulins. Neutralization is responsible for defending cells from pathogens, which are organisms that cause disease. Opsonization is a process by which a pathogen is marked for phagocytosis. Complement activation, key part of the innate immune system and plays an important role in the clearance of pathogens and apoptotic cells upon its activation. General functions of immunoglobulins.
Functions of immunoglobulin G, IgG.
IgG1, IgG3, IgG4, cross placenta and protect fetus. IgG3 activates complement. IgG1 and IgG3 binds to Fc receptor on phagocytic cells, monocytes and macrophages and mediate opsonization.
Functions of immunoglobulin A, IgA.
Constitutes 10 to 15% of total immunoglobulins. Present in milk, saliva, tears, mucus of respiratory tract, digestive tract and genitourinary tract. In serum exist as monomer. In external secretions exist as dimer called secretory. Immunoglobulin has J chain and secretory piece. Half-life, 6 to 8 days.
Functions of immunoglobulin A, IgA.
Provides local immunity. Secretory IgA binds to surface antigens of microorganism and prevent its attachment and invasion of the mucosal surfaces of respiratory and digestive tract. Immune elimination. Secretory IgA provides important line of defense against Salmonella, Vibrio cholerae, and gonorrhea, influenza virus and poliovirus. Secretory IgA present in breast milk protects newborn during first months of life. Activates complement by the alternative pathway. Promotes phagocytosis and intracellular killing of microorganisms.
Functions of immunoglobulin M, IgM.
Accounts for 5 to 10% of total serum proteins. Polymer of 5 monomeric units, pentamer, held together by disulfide bonds and J chain. Half-life, 5 days. Most of IgM, 80%, present intravascularly. Present in low concentration in intercellular tissue fluids. Cannot cross placenta. Presence of IgM antibody in serum of newborn indicate congenital infection. Earliest immunoglobulin to be synthesized by fetus, 20 weeks. First immunoglobulin to be produced in primary response tonogen. Relatively short-lived, hence its demonstration in the serum indicates recent infection. Monomeric IgM appears on the surface of unstimulated B lymphocytes and act as receptors for antigens.
Functions of immunoglobulin M, IgM.
It agglutinates bacteria, activates complement by classical pathway, causes opsonization and immune hemolysis, believed to be responsible for protection against blood invasion by microorganisms.
Functions of immunoglobulin E, IgE.
Structure is similar to IgG. Has 4 constant region domains, mostly present extracellularly, does not cross placenta. Produced in the lining of respiratory and intestinal tract, known as reagent antibody, does not activate complement nor agglutinate antigens. Different classes of immunoglobulins and its functions. Role of different immunoglobulin classes. IgG protects the body fluids.
IgA protects the body surfaces.
IgM protects the bloodstream. IgE mediates type I hypersensitivity. IgD role not known. Clinical use of immunoglobulins. Immunoglobulins are the antibodies produced naturally by the body's immune system, which help fight infection and disease. Immunoglobulins or antibodies can be used as a form of immunotherapy. Like drugs, they are prepared from a pool of blood donated at blood collection centers and processed through fractionation to separate the protein fraction from the cellular component. The purified immunoglobulin can treat many immunological problems. Antibody deficiencies, severe combined immunodeficiency disorders, SCID, multiple sclerosis, myasthenia gravis, Kawasaki disease, systematic lupus erythematosus, organ transplantations. Subcutaneous immunoglobulin therapy, SCIG, and intravenous immunoglobulin therapy, IVIG. Both IVIG and SCIG products are intended to get into the bloodstream. The difference between the two is that when IVIG is administered, there is a very rapid spike in serum IgG, whereas with SCIG, there isn't. The big spike from IVIG is often the cause of adverse effects that we don't see as commonly with SCIG. When given subcutaneously, Ig is taken through the lymphatic system and gradually enters the circulation, which results in a more consistent serum Ig level. Intravenous immunoglobulin therapy, IVIG. IVIG is a therapy used to treat antibody deficiencies related to a variety of inflammatory and immune-mediated diseases. It's made from antibodies called immunoglobulins, which your immune system typically makes to help fight off infections. This therapy can help people with weakened immune systems or other diseases fight off infections. Some of the diseases that intravenous immunoglobulin, IVIG, can treat include immune deficiencies like immune thrombocytopenia, ITP, Kawasaki disease, Guillain-Barré syndrome, chronic inflammatory demyelinating polyneuropathy, lupus, myositis. Other rare diseases, neurological diseases like myasthenia gravis or multiple sclerosis. People who receive bone marrow transplants may also use IVIG to prevent infections.